Okay, so before I dive into sort of the more details of multi-omics data integration, I'd just like to start by giving a very brief overview of what we do in the lab. Um, so in short, my background is quite diverse, and so our research topics that we pursue, but the umbrella that holds it all together is the application-driven development of trustworthy machine learning tools for large heterogeneous data. And we sort of aim at three different things in the lab. The first is we are developing tools and algorithms for analyzing imaging and molecular data. We work on the integration of multimodal data, both on the patient and the single cell level. And we pursue more theoretical work on uncertainty-aware and trustworthy predictive models, which is more at home in the machine learning and AI community. But today I'll really mainly talk about molecular profiling and sort of how we can integrate multimodal data on the patient level and also on the single cell level. And sort of our tools of choice are latent variable models. And this is at the heart of many methods that we develop and apply in the lab. And so what the idea behind latent variable models is that we start with a high dimensional data set of many correlated observations. So for example, we have like the expression of many genes. Maybe we measure also protein expression. Maybe you have the epigenome. Maybe we observe some mutations. And all of these features, especially within an omics layer, are highly correlated, of course. And if you were to look at each gene separately, that would be a very cumbersome exercise, especially if we have other omics layers as well. And so what we can do with latent variable models is to take this high dimensional space and map it into some kind of low dimensional data set of uncorrelated and hopefully interpretable or explainable latent variables. And so a very simple example of that is a group factor analysis that you may, or some of you may know in form of the MOFA model, where we start with a set of observed matrices where the samples here would be patients or cells and the features could be genes, and we measure gene expression. So each square here would be the expression of a specific gene in a specific for a specific patient. And maybe here we would measure the proteome and maybe the epigenome here. And then what we do is we take this matrix here, Y2, let's say the proteome, and model it as a weighted sum of some factors here. And now the trick is because we have several matrices that we have to use several weight matrices. So each of the patients all the patients share the factor matrix here. And to map the factor matrix into the observed space, we need to multiply it by sort of a omics specific weight matrix. And sort of what you can think of this as well is it's basically a PCA generalized to multiple matrices. And what you would see here would be the principal components or the latent factors, the PCs, and here would be like the loadings of your PCs. Okay, so you can do that, and that works quite nicely, actually, and you can sort of infer these factors. But actually, it's not so straightforward sometimes to interpret these factors, because sure, you can look at the weights, but it can become quite cumbersome. And I experienced that last in a project where we tried to analyze the proteogenome of AML patients together with Thomas Oellerich. There sort of was my first author paper, and so I had to do all the analysis myself, and we kind of inferred 30 different latent factors that were all explaining variants, and then I had to go factor by factor trying to figure out what the factor means, and then you do a gene set enrichment analysis and you try to map factors to pathway, and it's sometimes inconsistent, and it's a very cumbersome exercise. And sort of that motivated really what came next. And that was to say, okay, rather than doing this post hoc explainability where we fit these latent factors and then afterwards try to infer what they really mean, maybe we can build in interpretability in the model by design. Because we actually have a lot of knowledge a priori, even before we fit the model. We have, for example, databases of pathways where we know which genes or gene sets are associated to some kind of gene programs, maybe Reactome, KEC, or MSigDB. And maybe we can use this prior knowledge to link the latent factors that we learn to specific pathways. And that's what we did here in this movie model that we published a couple of years ago. It's led by Ava Koku, PhD student in the lab. And so the idea was here to come up with a Bayesian model where we use these gene set databases as a prior on the weights. So we basically argue that if two genes are annotated to a given pathway, let's say maybe cell cycle, and then these two genes are likely to have a high weight for that factor. And if a gene is not annotated to cell cycle, we believe a priori that this gene should have like a weight close to 0. And this is what we can model here with the so-called spike and slap prior, or here to make inference easier, we use a so-called horseshoe prior, which is essentially the same thing. But the trick is now that because this is a Bayesian model, the model can also refine these factor loads, and it can say, well, actually, this gene wasn't in the annotation, but it behaves very similar to gene number 3 and 4, which were annotated. So I'm going to switch it on and give it a high weight anyway. And by that, we can really learn these directly interpretable pathway factors and refine also the annotations and contextualize to a given context. There's also some nice theoretical properties such as identifiability, and we can make it all fast using stochastic variational inference. And I know this is maybe hard to get an intuition how it works, and that's why I have like a very simple toy data set to show or illustrate what this model can do. So what we did here was we ran movie on the TCGA data, pan-cancer data, where we had 4 different omics layers: DNA methylation, mRNA expression, microRNA, and proteomics. And we looked at 11,000 patients across 33 cancer types and fit our movie model. So that means we have to sort of annotate one of the data views and link it to some pathways. We chose MCDB hallmark pathways and linked or informed the mRNA data view with hallmark. And so then what you can do is you sort of learn the factors that drive the model, like your principal components. And the very first thing you want to check is whether these factors that you learn, which are now tied to pathways, capture the known global structure of the data. So just like you can run a UMAP or a t-SNE on the PCA, you can run a t-SNE on the movie factors that you infer. And what you would hope is that you capture the global structure of the data, which is sort of the different cell types. And in the t-SNE, you can see that all the patients cluster nicely by, by cancer type. What you can also see is that it's a bit tricky to interpret it beyond the clustering, because there's 2 types of skin cancer, this cluster here and that cluster here, which are both sort of on the outside, but really far away. So what does that mean? Are they really that different? Strange. And this is really why we'd like to have these interpretable models, right? Because with movie, what we can do now, we can directly look at the factor scores. And if we look at the one factor that we linked to the melanogenesis pathway that explained a lot of variants, we can see that patients who have skin cancer are clustering together here, and both skin cancer types have a high pathway activity of melanogenesis. And so this really gives a very interpretable latent space where you immediately can immediately know what it means if a patient has sort of a high factor activity in a certain dimension. And the same holds true for this androgen response factor, where prostate cancer patients have a very high factor activity and or pathway activity, so to say. Now, the other thing that you can do with movie is that you can really understand on a feature or gene level what these factors do, because you provide the model with these annotations, which are shown in black here, but the model can then decide which of these annotated genes have the highest weights and also switch on other genes. And so it can contextualize, in this case, the androgen response pathway to cancer, because you have a pan-cancer data set, right? And so you can see that the model also switches on other sort of prostate cancer related markers here, and we can also directly interpret which genes drive this factor. And another thing that we can do is that we can sort of look at other data views, because we've only informed the mRNA data view, but because the factor is shared across all data views, they can be interpreted via the same pathway. So we know that also in the proteome, this factor will explain sort of androgen response in the context of cancer. And when we look at the weights, we sort of can do some kind of are doing some kind of transfer learning to figure out or learn a protein signature of this pathway that we informed on the mRNA only. And if you look, this makes all a lot of sense, where we have like these sort of contextualized proteins on that other data view as well. Okay, so I hope that kind of gives you a little bit of an intuition of how the movie model works. And so next, I'd like to show what we do with that model in practice. And this is work where we work together with the Thomas Oellerich lab and the Lou Stouts lab and perform proteogenomic profiling of about 300 untreated DLBCL patients. And what we wanted to do here was to sort of infer the proteogenotypes within DLBCL beyond sort of the known main classification. And in DLBCL, patients are categorized into three main sort of subtypes. One is ABC, activated B cell-like, and germinal center B cell-like DLBCL, and UNC stands for unclassified. And so you have these three major subgroups that are known, but we hypothesize that there's more going on because there's a lot of unexplained heterogeneity in terms of outcomes if we rely only on these main categories. So what we did, we ran movie on these different data layers. So we had like the transcriptome, the proteome, and some mutation data view. And we informed it with 290 pathways from the NIH lymphoma signature database. And then movie identified, first of all, the major drivers of heterogeneity that's well known. So this classification that I've put in here again, this is called cell of origin, and movie learned sort of that there are 11 pathways that are related to the cell of origin. And if we make a UMAP based on all the factors that movie infers, we can see nice separation of the patients into by cell of origin. Now that's not very exciting because this is all well known, but the property of movie is that we understand exactly which factor does what. So if we want to discover variation between patients beyond what's known, we can just remove those factors or pathways that are related to the known classification of cell of origin, as well as stromal infiltration and proliferation, which are also known confounding factors that we're not really interested in. So we can remove these factors, then plot another UMAP, and then what we have here is sort of the structure or the secondary, let's say, structure of variation between the patients once we removed like these known factors of variation. We then could just do some graph-based clustering and identify 7 proteogenotypes, as we call them. We looked at these proteogenotypes in detail, and then I'd just like to very briefly highlight one of them, which is PG4, because we found that PG4 was actually a high-risk DLBCL. So when we performed survival analysis, we found that it had inferior overall and progression-free survival upon R-CHOP-based therapy. And importantly, it was independent of known risk factors such as cell of origin, genetic subtypes, lymphoma ecotyper, and IPI risk factors. And one reason for that is that it's actually by design, because we removed these known risk factor before we fit our UMAP and performed our clustering on the remaining latent space. We validated this as an independent risk factor also in several additional external cohorts. And because movie is interpretable, we can then easily also look into the model again and check which pathways actually explain these proteogenotypes. And in particular, PG4 was characterized by enhanced MYC activity and deregulated translation. So what I've tried to show in this application very briefly is that movie can disentangle this interpatient variability into these interpretable factors that are linked to pathways. It allows us to zoom into hidden variability and reveal new proteogenotypes in this case, just by removing sort of the known risk factors in the latent space. And PG4 that we identified has a low inferior survival in the entire cohort, also within known genotypes. So this holds true for MYD88 patients and double hit positive GCB patients as well, for example. Okay, so that's I think is quite a nice use case, and we were very happy with how this decomposition into the latent factors worked. And then we thought that maybe we can do something else with these type of pathway-based latent factors. And therefore, we teamed up with the Stegler lab at the University in Heidelberg, in particular with Florian Walter, Martin Roberg, and Ilya Katz from the Stegler lab. And together with those guys, Aber sort of modularized the movie framework and combined it sort of with a known MOFA code base. And we called the whole thing MOFA Flex because the idea is that we would like to, or that this will make MOFA, the original MOFA model, deprecated. So if you're a MOFA user, MOFA Flex will sort of be the next update, and it's already public. So I invite you all to try it out. And one nice property of MOFA Flex is that it's very flexible in the sense that it can also work with spatio-temporal data, for example. And the way we do that is that we say, okay, we can for sure place all these different types of priors on the weight matrices, like this prior knowledge module that I've just talked about. But we can also encode prior knowledge sort of on spatial coordinates by placing a Gaussian process prior on the factor scores themselves. And if we do that, we can, for example, jointly model spatial data like 10x Xenium and sort of high coverage non-spatial data like Chromium. And we did that as a sort of showcase here for FFPE breast cancer tissue, and leveraged the high coverage across the transcriptome from Chromium and inferred spatial activity of pathway scores using the 10x Xenium and via this Gaussian process prior. And without going too much into detail, what you can sort of see here then is that we can really infer explicitly these pathways that drive intercellular variation. And for example, the estrogen response early pathway then delineates cancer cells from normal tissue here, where you can see a high activity of this pathway and in areas that are cancer cells. And the apical junction pathway has a high activity for non-invasive tumor cells or cancer cells here, shown in this subplot here. Yeah. Another use case where we think it makes sense to think about pathways rather than latent factors in general is if we want to model regulatory effects of perturbations to genes or the drug response, because cellular networks or regulatory networks are very sparse and very modular. So if I perturb a cell and maybe I do some CRISPR knockouts or I put some drugs on the cell, then what I can assume is that these type of perturbations, these knockouts or drugs will affect only a very small number of gene programs. And very often what I'd like to learn is how can I quantify this effect in a meaningful and comprehensive way? So how can I learn which gene programs or pathways are affected by perturbations, and how can I generalize from that knowledge maybe even? And very naively, what you could say is, well, I have a drug and I would like to know how this affects a gene. So I just write down a linear regression. I do that for every gene. But obviously, then you will just end up with a large, very correlated matrix of effect sizes across perturbation and genes, and you have no idea how to interpret this. So other people have thought about this and suggested to say, well, let's work with latent factors and perform a latent variable model. Maybe in the easiest case, we take the expression count matrix and we perform a PCA, and then we have our latent factors, our principal components here, and we have our cells here. And then what we can do is we can, in a second step, fit this regression model and link sort of our perturbation design matrix to the principal components. So in other words, we'd like to use as input which gene was perturbed and use that to predict the activity of these latent factors. You can also do that not as a two-step approach, but sort of as an integrated approach and jointly learn this regression module and in the PCA module. But then still, it's very hard to interpret this effect size matrix, because you just know that given perturbations, let's say, have an effect on a module, but you don't really understand what the module is in terms of your pathways again. So, and that's where sort of our movie module comes in, because we can say, well, maybe we can again link our factors here to individual pathways via this horseshoe prior, and we can jointly learn this regression module together with these pathway factor scores. So in other words, what we do, we perform a regression that guides the pathway scores from the perturbation information. And the perturbation information basically just tells us for every cell how it was perturbed. So which gene was knocked out by a CRISPR, or which drug was given to which cell. Right? And so these are the inputs that predict the activity of a given pathway via this movie module. And we can also do that sort of in a multimodal manner. So we can write this down now again as our standard movie model here. So where we inform like the different factors, we make it multimodal, which has also not been done before, and we predict the pathway activity via this regression module from the individual perturbations. Okay. So yes. So first of all, what you did was to test whether that works and how well that works. And to this end, we compared PacMon, that's how we call our method, to GSFA, which is sort of the uninformed version, like this integrated PCA and regression module. And what we'd like to have is a very accurate reconstruction of, first of all, the factor loadings, so the weights of the genes, and also of the condition coefficients, so of the links between the individual perturbations and the individual factors. And we measured that by computing the area under the precision recall curve, and we found very high AUPRs both for the factor loadings, so the modules, how well the modules were learned, and also the condition coefficients, so how well the link between these perturbations and the individual pathways were learned. Another nice feature of PacMon is that it scales very nicely. So runtime stays very, very small, even for a very large number of cells or samples. Also, memory stays very low in comparison to GSFA, where runtime and memory explode if you go beyond like 10,000 cells. And GSFA also can only handle single modality data. So here's an example of how you can use PacMon. We applied it to a multimodal CRISPR perturbation screen of melanoma cells, where cells were cultured in 3 different conditions: control, indifferent gamma stimulated, and co-culture. And first of all, we want to again make sure that the factors we learn, the pathway factors we learn, are meaningful. So we hope that the cells would cluster in a global UMAP representation according to condition, which they do. We'd like to make sure that the individual factors or pathways they identify make sense, which they do. So here, interferon gamma stimulated cells have a very high activity in the interferon gamma response axis, and co-cultured cells have a very high cytokine signaling activity, which also makes sense. But that's all sort of like the basic movie stuff, right? So what we'd now like to see is how do perturbations link to these factors. And this we can also do, and we compute something called the PIP, which is sort of like a Bayesian version of statistical significance. If you have this significance, links are shown sort of with this black circle around. And the way to read this is that on the rows here, we have like different perturbations, and the columns are different pathways. So we now can check which perturbation regulates which pathway. And what you can see here is that the interferon gamma response pathway is, for example, strongly regulated by these perturbations. So if I have knockouts in these genes, then these will strongly deregulate or downregulate interferon gamma response, which of course makes sense if you look at interferon gamma receptor 1 and 2, for example, check STAT signaling and so on. We can do a similar thing. For example, we can look at EMT or TNF signaling via NF kappa beta, and then see which perturbations affect these pathways. And we can also nicely interpret whether there's an up or whether there's a down regulation in these pathways. And as before, we can also check what these latent factors really do, and can look at the loadings of those, as I've shown you sort of before. Because it scales so nicely, we thought we'd also have a go at the Tahoe 100 million data set, sort of this giga-scale single-cell perturbation atlas, where now perturbations aren't CRISPR knockouts, but they're drugs. And so what I'm showing you here is a subset of 1 million, but we also have now run it on the full 100 million data set. We just need to sort of update the results a little bit. And what you will get then is sort of, again, like this big matrix. And you're not supposed to read this. I'm going to highlight a few bits. But essentially, what you can see here is like a load of drugs. And whenever you see sort of like a dark color here, these drugs will significantly up or down regulate a pathway that is shown down here that we also learned with Pacman. And for example, what we have here is increasing doses, because in the tau data, you have not only different drugs, but also different doses. So you have increasing doses of Harrington-9, homo-Harrington-9, which all regulate TNF-alpha and TNF-kappa beta signaling, which correspond to the pathway shown below here. If you look at this group of drugs, these are doxorubicin, epirubicin, and corafenib, and sort of the pathway is hypoxia, and they all are known to regulate hypoxia. So we sort of recovered that as well. And also, when you look at this group of drugs here, these are dinacilib, axacimitib, citrate, and bortezomib. Sorry for the pronunciation. All of which have an effect on apoptosis. So we can sort of learn their mode of action by looking at these condition coefficients linking the drugs to the individual pathways. Okay, so that was all about like sort of this movie type modeling where we leverage these interpretability via these pathway factors. But of course, movie also has like one major drawback, which is that it's actually a linear model. And what we'd really like to have is a nonlinear version of that, but it should ideally be interpretable, maybe to a similar extent as factor analysis, which is of course impossible, but we still sort of had a go try to find that sweet spot. And what we did was to rely on Gaussian process latent variable models, which you can think of sort of a probabilistic and nonlinear kernel PCA or nonlinear version of a standard PCA. And it has two nice properties. One of them is it can be generalized to the multi-view case, so we can nicely analyze multi-omics data with it. And the second one is that we can make it interpretable via relevance maps, because we learn parametric mappings between the latent space and the observed space. And this has been around for a long time. Many years ago, we also, while I was at Helmholtz Munich, we applied it to single cell data at the time, which already worked quite nicely. However, it has one big drawback, namely that the standard GPLBM, so this underlying architecture, explicitly models sort of the correlation structure between samples or cells, but it pretends that all the features, so all the genes, are independent, which is of course not true, right? So to make this really usable for these complex multimodal data sets, we really want to make dependencies between the genes explicit. So in other words, we'd really like to learn the correlation structure also between the genes and not only between the patients or cells. And we can do that by learning latent variables, not only for the feed samples, so the cells, but also for the features, so the genes, let's say. And when we have a multi-view version, we can learn shared embeddings for the cells, and we can learn few specific feature embeddings. So we would learn, for example, gene embeddings for the RNA view and peak embeddings for the attack view if you have like a 10x multi-ohm data set, for example. Okay, so how does it look like? Here is like the classic PMMC use case of multi-ohm, and if you just run a PCA on this multi-ohm, you will see that you have like a very clear separation in three different broad cell types, so to say, but you use sort of a lot of resolution compared to the UMAP. On the other hand, for the PCA, you can nicely look at the loadings as you've done before, and you can sort of understand what PC1 and 2 mean in terms of like the loadings of the genes. Whereas for the UMAP, you're sort of stuck with the clusters and you don't really know what else to do apart from, comparing clusters. So in contrast with MomoGP, what we now get is, first of all, cell embeddings, which now come as a similar resolution as the UMAP rather than the PCA. And we also, in addition, learn these type of gene embeddings for the genes in this case. And what we can then do in this gene embedding space, we can compute sort of meta genes, which are sort of groups or modules of genes that are co-regulated. And then using the parametric mapping between spaces, we can check which cells in the cell latent space are most influenced by these metagenes. And then, for example, we can see that this metagene number 9 here has the strongest influence on PDCs. And if we look what genes these really are, then we found that these are really marker genes of PDCs. So we have like this really direct interpretability between these two different embeddings as well. And it also works for the multi-omic. So when you look at 10x multi-omic data, then we don't only have like the embedding of the cells and the genes, but we also learn this peak embedding. And again, we can sort of link the different embeddings via these relevance maps. There are also other methods which try to learn embeddings of also the features using graphs and neural networks, but they have two drawbacks. They're either not interpretable and or the embeddings, especially when it comes to peak embeddings, are not that meaningful because there's sort of just a lot of mixing going on here. Yes. Then I have one very briefly last little project that I'd like to present, and that addresses the question of how we can go from single cells to patients. Because of course, when we have when we're interested sort of in translating some of our findings, it's maybe not so useful thinking about cell types, but rather you want to think about patients, right? And so one solution is to go via tissues or cell type specific representations. So an idea that has come up recently was to pseudo-bulk tissues or cell types on a patient level. And once you do that, you can then disentangle tissue-specific heterogeneity across patients and thereby make this link between the single cells and the patients. So the way that works would be to say, okay, I have now cells from a whole lot of donors, and then for a given donor, I take all cells from a cell type and pseudo-bulk it. And then I can take this representation of my data and put it into a tensor, essentially. In my tensor, I have then my individual donors here as the rows. My columns would be the individual genes, and then my sort of third axis would be the cell types. So in each slice, I have then the expression of a given cell type, of a given gene for a given donor. And then I can do a tensor decomposition and sort of learn these latent factors that tell me how cells, how donors vary across cell types. Now we generalize that to the multi-omics use case, where we now not only have like one tensor, but we have multiple tensors, and they can even be of different orders, and we can perform this decomposition. And here's just a very brief example of how that works for the 10x multi-omic, where we looked at data from 18 pediatric patients with ALL and 5 healthy donors. And again, first thing we do is always to check whether we can separate the biggest source of variation, which would be healthy versus ALL, and that we can see very nicely within these two factors. You can see also the large spread in ALL compared to like the tight cluster of the healthy donors. And what we can then do is we can check how these different factors are driving variation for given cell types and given genes. And so what we can see if we look at factor 0 is that most of the variation is really driven by differences in blasts, which of course makes sense, and T and K cells, which makes sense. We can also look at other factors. We found, for example, factor 5 is quite interesting here because it shows like a small subpopulation within the ALL patients. And when we looked at the loadings, we saw that this was mainly driven by differences in PDCs, interestingly. And we're currently validating that, how to interpret that and whether that's a true effect. Okay, so I'm going to just skip that and summarize by saying that I've tried to show you how we can use interpretable latent variable models to infer unobservable hidden states from observable data by taking prior knowledge into account, and how we do that for the multi-view case, and then how we can discover gene programs that drive cellular heterogeneity and link them to perturbations. I've briefly talked about how to generalize it to the nonlinear case and for spatial data, and how to use tensor factorization to link single cell and patient level data. And with that, I just like to thank a few people, mainly, especially Ava Koku, whose ideas are driving a lot of this research, but also Sarah, Kevin, Sare, and Tira, as well as collaboration partners, the Stickel Lab, Hematology Frankfurt, and the Stout Lab. And thank you very much for listening.